Hello everybody. Today we will discuss about Notch signaling pathway. So this pathway is essential for lateral inhibition and cell fate determination. So let's understand what is lateral inhibition. So this is cell 1 and in close contact the neighboring cell is cell 2. Okay. Now this cell is developing into neural cell. But I don't want this cell to have a same cell fate, right? I don't want it to develop into neural cell. So there is a phenomena called lateral inhibition. So with the help of, I'll change the pen. With the help of delta plus notch, this will have lateral inhibition and it will develop into epidermal cell instead of neural cell. So this is cell fate determination. Now we have to understand how it occurs. Okay, so it's very important that we understand that these are EGF-like domains, which are very important for the specific interaction. So the first cleavage occurs in Golgi apparatus, and all cleavage are proteolytic. Okay, so this is the first site of cleavage. So it will create heterodimer 1, both are heterodimers, right? Heterodimer 1 and 2. So this segment will go and come in contact with delta and they will interact through EGF-like domains. This will go in endocytosis. Now these fragments are moving apart, right? This will expose the cleavage site too. Okay. So in the second cleavage occurs in extracellular domain. Now after second cleavage in the transmembrane segment, third cleavage will occur with the help of gamma secretase gamma secretase complex. So this is the complex through which your cleavage number 3 will occur, and there will be a notch tail after that cleavage. This notch tail will move into nucleus, will interact with this RBP-SUH protein, and this RBP-SUH protein will recruit co-activators, help in recruitment of co-activators and removal of co-repressors. Okay, so this is one thing that we need to understand. So therefore, the transcription inhibition or repression will turn into transcription activator. Okay, so this is regulatory sequence where it comes in attached. Now, yeah, so this is RBP SUH. It sits on the DNA and it is interacting with the co-repressant. So once the notch tail will arrive, it will displace the co-repressor complex and allows the recruitment of co-activators like MAML and histone acetyltransferase, turning into a transcription activator. Now, another important thing that this is a third cleavage, right? So it occurs with the help of gamma secretase complex. So an essential subunit is preselin. So if there is a mutation, right? So gamma secretase complex will not work. And if it is not working, then you just imagine that this is your tail. What you will do with this notch fragment? So there is no cleavage, there is no proper cleavage, okay? So you will generate extracellular peptide fragments which are basically, amyloid plaques. So you don't need those, what you will do with these undefined fragments. So this is also a frequent case for early onset of Alzheimer's disease. Next thing which is important is that Notch and Delta both are glycoproteins, and the interaction is regulated by glycosylation. So you add sugars to O-linked oligosaccharides, and this helps in increasing the Notch specificity. So therefore, we call it modulation of ligand receptor signaling. This glycosylation will enhance the Notch specificity. So this pathway is fundamental for animal development. The primary example is production of Drosophila